All right, well, we're going to continue here and we're going to begin looking at the actual um, steps, the actual phases, the events of meiosis. Okay, so just as a review, remember meiosis, what we're doing here, this is cellular division that is ultimately producing our gametes. So we're producing our sex cells. So these are going to be sperm cells in the male and the oocytes or the eggs in females, right? So this process, it's going to start off in a similar way to mitosis, which is cell division of our somatic cells, so all of our cells other than these gametes. And we'll kind of compare and contrast meiosis and mitosis here in just a moment. But we're starting in the same way in the sense that we're starting with that diploid cell, right? So in the previous video, we talked about the difference between something or a cell that is diploid and a cell that is haploid. So in this case, we're starting with diploid, and what we're producing are haploid daughter cells. Okay, haploid again meaning they have half of the necessary genetic material for a full cell, right, if you will, a full diploid cell. Okay, so again, these are going to happen in the gonads. So this is happening in the ovaries in females and in the testes in males. The gametes that are produced, the sperm and the egg, will have 23 chromosomes. Okay, they'll have 23 chromosomes. So the sperm will have 23. The paternal source, so the paternal gamete, the sperm, the maternal gamete, the oocyte, the egg, will also have 23. And in the event of fertilization, so when these two cells meet each other, when the sperm fertilizes the egg and their genetic material combines, now you don't have 23 chromosomes, but you have 23 pairs of chromosomes, right? So you have 46, right? And that's the first diploid cell that is produced, that's the zygote, okay? So what we're going to look at is the process of making those various gametes, okay? The process of making the sperm and the egg. So let me advance here. So mitosis and meiosis, quick overview, right? Quick overview in terms of the differences, right? So one is somatic cell division, one is sex cell division. So meaning, is this happening in our somatic cells? Somatic, that term soma means body. So think your skin cells and your liver cells and your cells in the kidney, right? All these other different types of cells that are not your gametes, okay? There's also a difference in terms of what you produce and the number of cells that you produce. So in mitosis, recall that you make only two cells. So you take one cell and you split that one cell into two. Those two cells are genetically identical to each other, and they're also genetically identical to their original parent cell, right? So you take one cell, split it into two, they're identical to each other and the cell that they came from, right? The reason they are identical is because you duplicate the DNA first, and that interphase process, if you remember from mitosis, we'll talk about interphase for meiosis here in just a little bit. You duplicate the DNA in that S phase, but when you duplicate the DNA, there's no intentional alteration of the DNA. So in meiosis, we'll have this process called crossing over. Okay, and we'll look at that in just a second. Okay, but in mitosis, genetically identical cells that are, you produce two of them. Right? Meiosis, you produce four cells. Those four cells are genetically different from each other, and they are genetically different from the original parent cell from which they came. Right? They are all haploid, meaning they contain half of the genetic material. They have 23 chromosomes, whereas the two daughter cells produced in mitosis are diploid. And again, the reason they are genetically different is because you have this crossing over. Right? So this crossing over is this process of the homologous chromosome, so meaning the chromosome from the paternal side, so from the father, and the maternal chromosome from the mother. There are exchanges, there are swaps of small segments of DNA that results in the two chromosomes now being slightly different than they originally were. So whenever they divide, when the cell divides and it moves these chromosomes and these chromatids into their respective cells, those chromosomes are ultimately going to be different from the chromosome from which they originally came, right? Where they came from. And so again, big idea with this crossing over is it contributes to genetic variation, right? It's contributing to the difference in the genetic material in the daughter cells compared to the genetic material in the original parent cell. Okay, so the first kind of event, the first thing that occurs before the actual meiosis is interphase. Okay, so recall interphase, this is like the preparation, if you want to think of it kind of like that. Big thing that's happening here is you are duplicating the DNA. The DNA in each cell or on each cell is replicated, right? So you're dividing it, right? Now, there's some terms that we need to be familiar with, okay? So these terms are chromatids and chromosomes. Okay, chromatids and chromosomes, and these are not necessarily the same thing. Okay, so first thing is a replicated chromosome. Okay, a replicated chromosome. A chromosome is a string of DNA, right? It's a strand of DNA, and so say you've got one strand. Okay, you take that one strand and you replicate it, you duplicate it, now you have a replicated chromosome. Okay, so that chromosome has two arms, right, two limbs, so these two here, one right here and one right here. So this whole thing is the chromosome, but you call each of these limbs of it, you call these chromatids. Each of these strands is a chromatid. Both of them together make up one replicated chromosome. Okay. You can also have homologous chromosomes, okay, or these pairs of chromosomes. This is referring to the same chromosome, so chromosome 1, chromosome 5, chromosome 15, doesn't matter, the same chromosome, but one from maternal, one from paternal. So one from the mother, one from the father. That's a homologous pair of chromosomes. Okay, so that's what we see right here. So this is replicated chromosome. This would be just maternal or just paternal. This is a homologous pair of chromosomes. Okay, so one maternal, one paternal, and then this here, this bottom one, is a single chromosome. Okay, a single-stranded chromosome. Right. So this here, this top part up here with the replicated chromosomes, each of these being called a chromatid. Whenever you split this apart, so you pull this chromatid to the right, you pull this chromatid to the left, you get this down here. You get now a single chromosome, right? So in other words, these sister chromatids in this replicated chromosome, once they're pulled apart, instead of calling them chromatids, now you call it chromosome, right? So all of this has to do with the organization, the arrangement of the chromosome slash chromatids, right? You're going to use different terminology depending on exactly how they're organized. So be careful when you are looking at this to make sure you use the correct terminology. Okay. So that's interphase, right? So kind of essentially getting ready. You're duplicating the chromosomes. They're going to have two chromatids, and this is necessary before we move into the events of meiosis, right? So interphase is kind of beforehand, right? Now we're getting into actual meiosis 1. Okay, so remember, this whole process, we're making 4 cells. Okay, we're making 4 genetically different haploid daughter cells. So we have to go through meiosis twice. So the first one is called meiosis 1, and then we'll go through meiosis 2. So for this first part of this video, we're going to look at meiosis 1, and then we'll do meiosis 2 separately, right? So this is the first event, meiosis 1. We've got our homologous pairs of chromosomes. So remember, homologous pair, remember, think one from father, one from mother. Okay, so one maternal, one paternal. We're going to stick those together, and then they're going to be pulled apart, essentially, into 2 cells, and that's meiosis 1. And then you're going to do that again, and that's going to be meiosis 2, right? So at the end of meiosis 1, we've produced 2 cells that are haploid. Okay, so at the end of meiosis 1, you have 2 cells that are haploid. Right. So let's look at the actual events. Right. So first is prophase 1. Okay, prophase 1. So prophase 1, you have the homologous pairs of chromosomes. So these homologous replicated chromosomes. So one maternal, one paternal. They're going to come together. They're going to move together, physically together, and they're going to join. Right. They're going to join at a region called the centromere. Right. So when this occurs, and you have the maternal chromosome, with its two sister chromatids, so say this is the red one, and say this blue one is the paternal chromosome with its two sister chromatids, they're going to join together and form something called a tetrad, right? A tetrad. This process of these two coming together is called synapsis. So the result of synapsis is the formation of a tetrad, right? So the tetrad is the maternal chromosome with two chromatids, the paternal chromosome with two chromatids, both of those stuck together, right? That's a tetrad. Okay, this will be important in a moment, right? We'll talk about why this is occurring. During prophase 1, you also have something known as crossing over. Okay, so crossing over is essentially exactly what it sounds like. You're taking a piece of the chromosome on each one. So the maternal replicated chromosome and the maternal, so one from the mother, one from the father, and you're swapping or you're crossing this piece of the maternal chromosome with this piece of the paternal, right? So you're exchanging genetic material, right? One part of the maternal is being swapped and replaced with one part of the paternal. Okay, so this kind of illustration down here, you've got paternal and maternal, they swap a little piece and now they're different, right? So now automatically we can see how our cells that we started with these chromosomes, we're going to have different cells that we end up producing at the end of meiosis 1 and at the end of meiosis 2, right? So again, big thing, genetic variation. We're producing different daughter cells. Okay, remember, that's the whole point. That's the whole point of this whole process. Okay. So again, crossing over, big idea, right? Big idea. You're also having microtubules extending from the centromere, going to these replicated chromosomes and attaching to them at this region called the kinetochore. So this kind of midline region near the centromere, and this is ultimately going to allow them to be separated during metaphase. Okay. So speaking of which, metaphase, when you think of the word M, think middle. Okay, so M in metaphase, think middle. The replicated chromosomes here are going to align along the middle of the cell, along something called the metaphase plate. Okay, so they're going to align along this metaphase plate, and one chromosome with its sister chromatids is going to be on one side. So in other words, facing one pole of the cell, facing one side, and the other chromosome with its replicated one replicated chromosome with its chromatids is going to be facing the other side of the cell, right? So the side that the chromosomes face, whether they face the left side of the cell or the right side of the cell, it's random. Okay, so this is called random alignment. And then there's something called independent assortment. Independent assortment is meaning, okay, so say for chromosome 1, the paternal, so the chromosome from the father, chromosome 1 from the father, is facing the left, all right? Chromosome 2 from the father may not be facing the left. It may be facing the right, or maybe it is facing the left. It's random. They align themselves independently of each other, okay? So meaning paternal chromosome 1 faces the left, paternal chromosome 2 faces the left, paternal chromosome 3 faces the right, 4 faces the left, 5, 6, and 7 face the right, 8 faces the left. It's random. Okay, they align themselves independently of each other. Okay, so this is referred to as the law of independent assortment. Right, they're going to be moved to one side of the cell. They're going to end up in different cells. Right, again, contributing to genetic variation. Right, you've also got those spindle fibers again. They're continuing to attach to the kinetochore of the chromosome, right? And those chromatids are then ultimately going to be pulled to opposite poles of the cell, right? Ultimately pulled to opposite poles of the cell. So here you can see those cells aligning along that metaphase plate. You can see the kinetochore, right, as a part of the centromere, and you can see those spindle fibers reaching out and attaching to those. Notice also how you have the different replicated chromosomes facing different sides. So let's say the red is the maternal from the mother. Let's say this one is facing this side of the cell. This maternal chromosome is also facing this side of the cell, but this red one is facing this side of the cell, right? They align themselves independently of each other. They don't all have to be facing the same direction, right? So we're aligned along this metaphase plate, and now we're going to begin pulling them apart, right? So focus on this top graphic here. What we're doing is we're taking those spindle fibers, those microtubules that are attached to the kinetochore, and they're going to be pulling and separating those replicated chromosomes, right? So again, these are homologous pairs of replicated chromosomes. So again, one is from maternal side, one is from paternal side, and they're going to be moved to opposite sides of the cell, as you can see here, right? So here they're aligned in the middle. They are attached by the kinetochore, or the microtubules to the kinetochore, and then they're being pulled to the opposite sides, right? Notice here also the genetic variation, right? So crossing over has already occurred during prophase 1, as we've mentioned, right? So this is called reduction division. Reduction meaning we are reducing the number of chromosomes that are going to end up in each cell, right? So remember, we started off diploid, and we're ultimately going to end up haploid, right? So here, in a single cell, when you have maternal and paternal, this is diploid, but whenever you separate them, now you only have one or the other. It's haploid, okay? So this haploid is going to result in only half of the genetic material ending up in each cell. Okay. So this last phase is going to be telophase and cytokinesis. Right. This is essentially resulting in the chromosomes aligning or ending up on one side of the cell, so one pole of the cell. And again, each of those cells is going to have 23 chromosomes. Right. Each will have 2 chromatids, but remember, it's just for one replicated chromosome, so they're technically haploid. Okay. And then cytokinesis is the process of essentially just splitting those cells in half. Okay, so this last graphic here is showing telophase and cytokinesis. So you're bringing in these actin protein fibers, and they're essentially pinching off these 2 cells and separating them. And what you've produced now are 2 haploid daughter cells. So meaning 23 chromosomes, as opposed to our original diploid that had 23 pair, right, or 46. Okay, so we're going to stop here, and we'll pick up in our next part, and we will look at the second half of meiosis, called meiosis 2.